actual title is kind of long, so I summed it up by saying multi-dimensional entourage effects. I'm sure everybody here has heard of the entourage effect and maybe even know something about why it's so important in cannabinoid therapeutics. What I'm going to do is try to expand your vision of the entourage effects so that it's in many different dimensions. And the reason, as you'll see, is because the endocannabinoid system and cannabis, the plant that produces the natural substances that bind to the cannabinoid receptors, is really not a simple one or two molecule business, but hundreds of different molecules. And it's because they can be used in different combinations, different ratios, et cetera, even different means of ingestion, that we find it has therapeutic properties across a very large swath of medical conditions. And this is very often used as an argument against medical cannabis, saying how could something be effective across so many different conditions when, you know, we live in an age of very specific targeted therapies for things. And I think it's through understanding how special the entourage effect can be once you broaden it to consider all the different types of compounds and receptors that are involved, they can get an idea as to why there's a potential within the plant to treat so many different problems. My own career comes from that. I started out with an MD PhD focusing in lipid biochemistry, which led me to psychopharmacology research. But then along came the AIDS epidemic, and I found myself very much involved there and studying the role of recreational drug use in transmission and development of immunodeficiency. And that's where I really learned that cannabis was different than all the other recreational drugs that people were at risk or had HIV. It did not help spread the virus, and it did not have any negative effect on the rate of development of immunosuppression. If anything, patients and others were reporting to me the use of cannabis to treat the side effects of the medications or some of the major effects of AIDS before we had treatment, wasting, cachexia, etc. So it seemed very different than all the others, and it led me to become an activist in the medical cannabis scene, and I was amazed at how few of my colleagues, professional colleagues, who were very open to ideas in other areas, had very closed minds when it came to medical cannabis. And so that's another very important thing to keep in mind here, that whatever we're talking about medical cannabis, we have to, you know, consciously take into account the stigma that's there, both against the plant and its products, against the patients who use it, and even against researchers and clinicians who are trying to further it, because the standard answer is, well, where is the data? Where is the proof? And even after you explain to people that prohibition has kept us from getting that data, they still insist on seeing it before they're going to change their mind. And a lot of that has to do with 70 or so years of prohibition and a lot of propaganda against the plant and its use. And I think some of that continues to this day, although a little subdued, but it's still there nonetheless. So when I retired from clinical work, I became pretty much a full-time advocate for medical cannabis research, helped develop a system for doing community-based, patient-centered research. which I'm still working on getting going. But within the last year, I found Nirvana, which was a group of people in Jamaica who had already started developing a canna resort there, Coral Cove, and were very receptive to my idea of expanding it into a therapeutic center where people come down and get treatments, where we could develop new treatments, but even more so we could educate healthcare professionals in how to integrate it into them. So if you want to know anything about Coral Cove, I left some brochures in the back there on the table, and I'd be happy to answer questions or give people my cards about it afterwards. It's being constructed now on the site of the old resort and should be open for the first patients in April of this coming year. Okay. So why is it the entourage effect so essential to understanding and developing therapeutics? Well, a lot of people think it's just you have two receptors and you want to balance the amount of activity in the two so you get a balanced cannabinoid response. But that would be very simplistic if we just stopped there, and in fact wouldn't even be totally true, because most people think of phytocannabinoids as the main ones being THC and CBD, so they automatically think since THC binds to the type 1 receptor, that CBD must be a specific ligand for the type 2 receptor. And it turns out that's not the case at all. In fact, CB2 is a allosteric inhibitor of both agonism at A1 and A2. So it acts as kind of a brake on the effects of alpha-1 agonists. delta-9 tetrahydrocannabinol. So it's very important for that first level of the entourage effect. But it's only the first level. This is a slide from a friend of mine, Dr. Rita McGuire, which really tries to get the point across that there are so many different physiological systems in the human body that have either been shown to be amenable to treatment with cannabinoids, or there's gathering evidence that they are a target of cannabinoid therapy. And so as soon as you start talking about, say, a single drug that would hit all those physiological systems, it starts to get kind of hokey, doesn't it? Like you're selling snake oil. And that's because we're not talking about a single drug. We're talking about a drug that can be individualized into hundreds, if not thousands, of different specific makeups, what particular cannabinoids are in it, and you'll see other molecules as well. So this is a slide that I want you to commit to memory, because there'll be a test on this slide at the end. No, I'm teasing. It just lists the fact that there's another very important aspect that is part of the entourage effect, and that's what we call allosteric interactions. And that is, even if the cannabinoid is binding to the CB1 or the CB2 receptor, it can have effects on other receptors that are nearby. And the classic one that I'll get to is the opioid receptor. The cannabinoids do not bind directly to the opioid receptor, but they have this allosteric effect where if you use the right antagonist and change the conformation of the cannabinoid receptors, it has an effect on the opioid receptors, which makes them much more sensitive, so you use less opioid to get the same analgesic effect, and other effects that are very, very important in the goal of using cannabinoids to cut back on opioid dependence and to actually treat people who have gotten dependent on high doses. So we're going to go through a lot of different examples here just to show how all these different types of receptors might be involved in the effects in different illnesses, different situations. But you see at the top, you have the classical endocannabinoids, the anandamide and 2-EG. And those are clearly type 1 and type 2 agonists. But as Dr. Mechoulam points out, nobody's really studied the effect of giving humans endocannabinoids. You know, it's all cannabinoids. No one's really, we haven't gotten to the point where we are manufacturing and giving people the actual substance that their body might be short of. And then there are other peptides that will attach to the CB1 and CB2 receptors and change their affinities and so forth. And then there are these substances, which are basic phospholipids, which also can have effects on the classical receptors. But then you start having the fact that each of the classical receptors can be associated with other receptors, what we call metabotropic nuclear, meaning they're receptors that change the transcription of DNA to RNA in the nucleus, so they change the expression of proteins, okay? And ionotropic are ones that change the cellular membrane channels through which ions pass. And we know that in many systems, like particularly the nervous system, it's those channels and the changes in the concentrations of specific ions that is greatly important in changing the sensitivity and the ease of discharge and so forth of those particular neurons. And then when you get down to how these compounds are being metabolized, then you have the ability to think about, well, maybe instead of changing the amount of a particular substance that we give a person, we change the rate at which it's synthesized or degraded. That has not been a very useful method, because many of these enzymes are relatively non-specific, so if you go to inhibit them or enhance them, you find a lot of undesirable side effects. But there has been one very interesting thing, the FAAH, which is so important in the metabolism of anandamide. Recently, you may have read about the people who don't feel pain, and they have a genetic defect in the FAAH gene, so their pain receptors are constantly bathed in anandamide, and so it's as if you're giving them a huge amount of an anti-pain pain-disrupting compound. And the other interesting thing is not only don't they feel pain, but they get, sores and cuts and burns and things, in part because they don't have that sensitivity, but they seem to heal much faster. So there's lots to be learned yet from these pathways and how they might affect some very basic cellular physiological activities. This is like a summary slide that gives you an idea of what's going on whenever you stimulate the cannabinoid receptors, okay? So that's what agonism means. So it's an agonist that when it binds the receptors, turns them on and produces whatever effect is going to happen. And as you can see there, not only are there the CB receptors types 1 and 2 illustrated in the middle, but there's other receptors, in particular the TRP, or a vanillin receptor, and the VGCCs, and those affect the level of calcium in the cell. When they are changed by binding at the CB1, CB2 receptor, they allow calcium to enter the cell, and that has an enormous effect on the cell potential and the ability of the cell to function. So as you can see, that at the top you have all the different effects agonism of the cannabinoid receptors could have, but those effects then go through either directly or via a second level of a messenger, and eventually get down to the most basic cellular physiological processes possible. On the left you have a stylized mitochondria. There's many diseases which are turning out to be defects of how energy is produced and metabolized in the cell, and it seems that the cannabinoids affect that to bring it under control. Then you have the nucleus, and I talked about how we have changes in what is being translated in the nucleus and what proteins are being produced. And that calcium affects the entire endoplasmic reticulum system, where these proteins get produced and packaged and put into place. And you see cell fate, ultimately, whether the cell remains in steady state, it is determined by a number of processes. The metabolism, there's another system called the ceramide system, where the amount of concentration of ceramide in the cell tells the cell whether it should divide or stay as one cell, and so forth. And what do you get when this whole system regulating cell stability and multiplication goes awry? What disease is characterized by that? Cancer. I mean, that's cancer. When the normal rate of cellular division to replace cells as they die gets out of balance, and a set of cells starts reproducing and dividing and is not under the normal controls and just pushes aside other cells, that's cancer. So if I had a slide to show you, I would say that the bottom line for all of this is that the entourage systems exist to maintain homeostasis in any physiological system. And that can be at the level of a cell, it could be the level of a tissue or a whole physiological system, or of the whole organism relative to its environment. And homeostasis is defined as a dynamic process by which an organism maintains its internal environment despite disturbances from external forces. And that could be said at the cellular level as well. So that's really what the bottom line of all these effects is about. We see this happening at almost every major physiological system that requires homeostasis, that requires to be kept in a narrow range, where if there's not enough of it, there's a deficiency of that system, and if there's too much, then there's problems because of hyperactivity of that system. And the best example of that is the inflammatory system. We all know that the purpose of the inflammatory system there is to remove dead cells, remove foreign cells, viruses, etc., that don't belong there. The problem is, once you start stimulating the inflammatory system, it can get out of hand and can actually attack the body and its own what should be there, its own proteins and so forth. And so you need a system whereby the inflammatory system can be selectively antagonized and brought up to hit a threat, and then can be brought back to homeostasis afterwards. And that's one area where we know that the cannabinoid system is very much involved, because that's where we have so many of both CB1 and CB2 receptors in the inflammatory system throughout. In the brain, we have receptors on glial cells, and glial cells, as you know, are the nourishing cells, the protective cells that protect the neurons and so forth. So that's why we're finding that certain cannabinoids can protect the brain and parts of the brain even after trauma or a stroke or whatever, can prevent cell death. Within the neurons itself, it can change the excitatory nature of the neurons, and we see that when we use CBD and other preparations to treat epilepsy. You're bringing down the excitability of the nervous system. All right, so here is an example I was just talking about. How do cannabinoids affect the glial system and thereby affect the integrity of the nervous system? So, as you can see here, a lot can happen when you go from a resting microglial cell to one that's activated or stimulated and then needs to be downregulated. And you see how not only 2-AG, but also these other similar compounds that are found in the human body, but are not endocannabinoids themselves, these resolvins. And then we've already talked about that. We've talked about immune cell maturation, that the depending on whether it's CB1 or CB2 being stimulated, you get different cell types of the immune system turned on and off. And eventually, if we figure it all out, we can figure out the role of all these different compounds in controlling just what immune cells are activated under what conditions. And that would be an enormous boost in many areas of therapy. And one of the central targets in all this is a translational factor called NF-κB, and as you can see, when that's turned on, it's on there. It's up. Don't do anything.
I'll hold it like this the whole time if you'd like.
Okay. When that can control all different cell types and cell processes in the immune system, including cell death and control of cancer. Now, I want to skip ahead since we've lost some time here and just mention that very recently we've seen that certain aspects of the cannabinoid system, the ECS system, can actually have effects on the accumulation of the synaptic tangles that are thought to produce dementia, in particular Alzheimer's. It's a very early stage disease, but people are working on different analogs of cannabinoids that might actually not just stop the accumulation of these amyloid tangles, but maybe even prevent them or reverse them in people who started to have early dementia. So that would be open up a whole nother area of research and therapy based on this multidimensional system. I was going to go to the next. I'm just going to use there we go. So that shows you the basic outline of what's going on with the amyloid system. And here's a very important slide. I talked earlier about allosteric effects, and this just illustrates how CB2 agonists and antagonists can have effect on another receptor, the the CPR55 receptor, okay? And they can work together to have cross agonism, or they can work to get cross antagonism, or one could actually move the other one in the opposite direction. So, and this is the basis of a lot of the effects of cannabinoid as therapy in pain and opioid dependence. Because what we find is that the opioid receptors that are using the opioid compound to produce analgesia, that their sensitivity to the opioid agonist is changed by binding to the associated cannabinoid receptor. And then we have this whole other system, the mentioned before, the vanillide receptors, and you can see here in these mouse models that different ways of stimulating those receptors, and it can be blocked by using cannabinoid blockers, so they are linked, can express very different processes in mice, and there's a huge amount of research going on to see how much of this is relevant to the human. Part of the problem is that a lot of these studies are done by using chemical analogs of naturally occurring cannabinoids. So you want to have something that will block, say, a particular receptor or block a particular cannabinoid agonist. And those synthetic ones are, again, can be very nonspecific, they can be very potent, and they can have, you know, unexpected adverse side effects in humans. So this is an area of research where it's moving ahead, but very slowly as we get to better understand how to make the effects specific at any one particular level. And you can see here that when it comes to the TRP receptors, it's incredibly complex. And they can have effects at all different levels, from the plasma membrane, changing the level of the calcium coming in, which turns on and off the various kinases that turn on and off of the processes subsequent to binding, but all the way down to the nucleus where genes are activated, which then produce pro-inflammatory or anti-inflammatory substances. So it's really tremendous. And that's all been talking about cannabinoids. Now I'm going to really blow your mind and bring in terpenes, okay? And in fact, in doing the literature search for this, I saw that one of the first papers on the entourage effect was actually by Aaron Russo, and it wasn't about CB1 versus CB2. It was about cannabinoids and terpenes. That was a very important entourage effect that needed to be looked at. So, for example, in our work at Coral Cove, we'll be looking at pain and how to treat it with cannabinoids. We don't want to forget that there are some specific terpenes, like in the middle there, myrcene, that are well known to increase the analgesic effects of other substances, including cannabinoids, and also to help the person feel less anxious and depressed and so forth. We know that has an effect as well. So there are some aspects of this complicated, multidimensional entourage effect system that we're ready to start using right now, because substances like myrcene and the other terpenoids, they've been around for decades. They're food additives that are very well known for their therapeutic uses and so forth. So there are no real problems introducing these into the mix when we go to treat or develop treatments. And then We think we have the whole universe, and then we start finding out that there's lots of other compounds that we didn't think much about. So, for example, CBG's been around for a long time, but most strains of cannabis when grown don't have much CBG because it gets converted. It's the mother compound from which all the other cannabinoids are made. But there are now people who are developing strains that end up with very high levels, 10, 15, 20% CBG, making it practical to isolate and test that. And it has its own anti-anxiety and anti-inflammatory effects. We talked about CBD being an allosteric inhibitor of CB1, CB2, but it also has anti-inflammatory and anti-anxiety effects. And if you look into that, you see it's through the GABA receptor. And the GABA receptor, as we know, is where anxiolytics like diazepam work, okay? So we may someday be using CBD instead of benzodiazepines to treat anxiety, or some combination of CBD with other compounds. And then we have other minor cannabinoids. I just listed a few of them that are beginning to get studied as the genetics and chemistry and so forth of handling them. Like Meshulam's lab has now found ways to stabilize the acid analogs of THC and so forth. Another lab is using THCV for its anti-pain, anti-inflammatory effects, and CBN has yet to really come into its own in this area. And then just last couple of months, Meshulam's lab announced that they've discovered a whole new set of compounds produced in the body. They call them anandamide-like molecules because they have the main structure of anandamide, but then instead of the alkyl group added, they have an amino acid group added. And these are specific compounds that your body produces in response to stress to limit the effects of the stress on that system or to undo it. So, for example, one of them will restore bone density in an animal model of osteoporosis, and the one that I'm most interested in will undo the effects that addiction has on the synapse, which is something that remains even after you've gone off the addictive substance and causes cravings and thus leads to relapse and really is a limiting factor in treatment. So I just want to thank my research assistant on this and all the people whose work has come before that's made this possible. All right, well, thank you very much for sitting through all that. And I really apologize for the problems with the slides, but if anybody is really interested, they give me your card. I will be happy to send them the slide deck. Thank you.